Hello, my name is Jean-Luc Fraikin. I'm CEO of Spectradyne. Thanks very much for tuning into my talk today. You've probably heard of us for EV quantification technology, but today I'm going to describe how microfluidic resistive pulse sensing delivers accurate and rapid measurements of virus concentration instead. This is far faster than live biological titer can. Let's start by reviewing why virus concentration is important in the first place. And the reason is basically because virus concentration is a critical experimental variable, and when you control it, you can have tighter experimental outcomes. The value of this really shows up in two key areas. First, when we're using virus as a therapeutic, so obviously gene therapy, for example, in these situations, concentration of the virus equals the dose of the drug. So it's a critical experimental variable, obviously, and it's important to control it for safer, more effective products. Second, virus being used as an assay readout. And in this case, so for example, diagnostic testing, in this case, the concentration is the test result itself. So the better you can measure it, the higher sensitivity and specificity you will have in your assay. So the bottom line is basically virus concentration is a critical variable that has to be controlled. And when it is, we end up with tighter experimental outcomes, faster science. So how is viral titer usually obtained these days? Well, the gold standard is live biological titer. This is a tissue culture technique. And if you've ever done it, you know how painful it is. At best, it takes hours to get your answer, but it can take as long as days. It's very error prone, requires many replicates as a result. And just the whole tissue culture infrastructure presents an enormous time and physical dollar cost also. So we want to try and do better than this. Optical particle analysis methods, these are challenged by this application because viruses are weakly scattering biological nanoparticles, just like EVs. So not to mention, they live in polydispersed complex samples that easily overwhelm optical methods, just like EVs. So we need better than that too. And here's where microfluidic resistive pulse sensing, or MRPS, from Spectradyne comes in. It delivers fast and accurate concentration, and it's affordable and easy to use. Our technology comes in the form of the NCS1. It's a benchtop instrument that delivers accurate concentration starting around 50 nanometers diameter and up. It uses electrical detection, not optical techniques, to measure the particles. That's important. And it leverages everything there is about microfluidics that makes it so exciting. It's easy to adopt. And because the microfluidic cartridges are single-use disposables, they're contamination-free. You don't have to clean anything in between measurements to avoid cross-contamination. Compare that to working under a Bunsen burner, for example, to do phage titer. One other bonus is that there are only 3 microliters of a sample required for analysis. Let me give a quick plug for an upcoming webinar that will be hosted by Select Bio that we will sponsor. It's called Fast and Accurate Virus Quantification, including SARS-CoV-2. This will take place on Tuesday, February 9th at 11 a.m. Eastern Time. And we will have Dr. Zoltán Varga from the Research Center for Natural Sciences in Hungary speak and describe his measurements of SARS-CoV-2 using MRPS. You can register either by emailing us to get the link or to follow this registration link to go to Select Bio's registration page. Let me briefly describe how the NCS1 works in case you're not familiar with it. The technology is called microfluidic resistive pulse sensing, and particles are counted one by one in this technique. Concentration and size are measured as directly as possible. What we do is we flow all the particles through a bottleneck in the fluid flow constriction, and every time a particle passes through that bottleneck, we get an electrical signal that looks like this on the right side here. So we measure 2 things about it: how big it is, that tells us how big the particle is, and then how quickly the particle went through the constriction tells us the volume of sample that we've measured. And so with counts and volume measurements, we can determine concentration directly. So here's a longer measurement showing many particles being detected. You can see that there are a few different sizes of particles in here. You can read that directly off the histogram. And when we convert counts to concentration, the output looks like this. So this is a plot with concentration on the y-axis and particle diameter on the x-axis. You can see the sample contains 4 different populations of particle sizes. This was just a mixture of polystyrene beads, 50, 90, 120, and 150 nanometers in diameter. And this combination of accuracy with ease of use is just not available with any other method at this point. We've got lots of literature to help convince you of that. Let me show you some measurement examples, but first I will give a plug for Dr. Zoltan Varga's talk in this session a little bit later at ISEV ID. He'll be describing briefly the measurements of SARS-CoV-2 using MRPS. That will be at 2:50 p.m. Eastern, and please tune into that. And if you want to learn more, join our webinar too. So the first example I will show is a measurement of human adenovirus 5. And I wanted to start by just showing you what the raw data looks like. So what we have is a baseline here, and then every time there's a particle go through, we get a spike, an upward spike, and they're highlighted by red dots, just like I showed you before. So here we're counting virions one by one. You can see your virus, right, over time being detected. Now, when we convert that to a concentration, particle concentration versus size plot, this is what we get. In this case, I'm showing you 3 replicates of the same sample of the adeno5 virus. And we can see there's a clear identifiable population from the virus in the sample. There's high repeatability between the replicates. And this measurement is quantitative over any size range. So, of course, you're interested in measuring the concentration of the virus, but this technology also allows you to quantify off-peak particulates. So that can be important depending on the application, which I'll describe in a minute. So when we measured just the concentration over the size range of the virions, we see very accurate reproducible measurements, good replicates here. And just as a reminder, this is absolute concentration in about 5 minutes per sample with no tissue culture required. So this was a super easy, 15-minute measurement set. Let me describe what you can do with this technology. So the first example is to assess purity of a virus preparation. So in this case, this is a commercial customer that is preparing viruses, and we measured 3 different stages of purification. First was the crude preparation in dark blue at the bottom, and you can see there's no peak in the size distribution. There's a slight peak emerging after first step of purification, and then after purification number 2, you can see that the virus has been enriched and now stands out over the background in this sample. And so this is incredibly important to this customer because it allows them to tweak parameters of their purification processes to optimize the production of their material. Finally, I want to highlight another key application of this technology, which is because the measurements are so quick, it allows you to gain essentially real-time measurement of virus concentration for optimizing production. So this measurement here is showing you crude Myxoma virus preparation. And you can see the sample contains a broad background of random particles, probably cell culture debris and proteins. But there is a clear signature from the Myxoma virus right where we expected, around 260 nanometers diameter. When we zoom in on this, you can see there is a clear peak, and we can quantify just the virus in this sample as being 2 e to the 10 or 2 e to the 8 particles per mil. So what this allows you is in just 5 minutes to take a sample of your production sample as you're producing the virus and measure the titer in a few minutes, and then wait until the concentration is optimal, maximized before harvesting your virus sample. So this is incredibly powerful value in this application. Finally, I'll show you a couple of slides describing the performance of the technology to you. First of all, first example shows a retroviral drug formulation, a serial dilution. You can see the viruses are showing a clear peak in the distribution here. And as we plot the concentration as a function of dilution factor, we get a very excellent linear response. Lastly, I'd like to show you about the dynamic range of the technology. This is a measurement showing the measurement of a single sample ranging from about 65 nanometers up to 5 or 6 microns in diameter. And you can see the concentration in this sample spans over 9 orders of magnitude within a single sample. And there's just no other technology that can currently deliver this kind of measurement. Our customers measure all different kinds of samples, different viruses. If you don't see yours on this list, then email us to learn more or send us a sample so we can show you what it looks like in high res. In summary, Spectradyne's technology delivers accurate viral titer in just a few minutes per measurement without any of the hassle of cell culture. And this allows you to do better science faster, which is great. Please contact us to learn more, and we love demonstrating our technology, so send us a sample to see for yourself. Thanks for your time again, and I hope you enjoy the rest of the conference.